For this assignment, envirocare created an SOP by using and collecting samples from a designated area at Texas A&M University. The purpose of this standard operating procedure is to describe procedures for the collection of representative liquid samples from a stream located near the golf course. These samples are collected from the surface and near the surface of the water. In order to collect these samples, a dipper and a bailer is used.
Sampling with a dipper. Once your site is prepared and your sample sites are chosen, make sure your sampling equipment is clean and ready for use. Begin at the sampling site furthest downstream. From a safe distance, use the long arm of the dipper to collect water in the sampling cup. Take care not to disturb underlying sediments to prevent contamination of your sample. Using a cleaned X-Tech water multimeter, record the sample's conductivity, salinity, total dissolved solids, and temperature. Using the HANA pH meter, record the sample's pH. Pour the sample in the dipper in a clean sample jar, being careful to avoid spills and contamination. Seal the jar and properly label it according to your chain of custody plan. Place the sealed jar into a cooler and store at 4 degrees Celsius. When you're done, rinse the dipper, X-Tech water meter, and HANA meter with distilled water to prepare them for their next use.
Sampling with a bailer. A bailer is applicable in a sampling site with a high distance from the water, such as shown. Ensure that the bailer is cleaned and functioning properly. Begin by passing a string through the bailer's handle. Ensure that one end of the string is anchored and allows the bailer to reach the water. In this case, we used a second team member to hold the string. Remove the tube attached within the tip of the bailer, allowing water to flow into the bailer's holding cylinder. Feed line slowly towards the anchored end to drop the bailer into the water and collect the water sample. Note: ensure the bailer only samples the water's surface and does not sample the bottom of the sampled water body. Withdraw the bailer from the water, ensuring the ball of the bailer seals the water sample within the bailer. It is vital that the bailer stays as vertical as possible to ensure the ball seal functions properly. Insert the tube into the tip of the bailer, thus releasing the water sample into the sampling container. Note: ensure that PPE is used and functioning properly, as contact with water sample is possible in this step. Properly label and seal water sample, then place it into the cooler.
Plating procedure. It is necessary to plate the water samples to observe bacterial growth. This can be achieved by abiding by the following procedure. 1. Begin by using serial dilution to achieve the proper dilution of your sample for plating. 2. Measure out 9 milliliters of pure water, then add 1 milliliter of sample for a 1 to 10 sample dilution. To prepare a 1 to 100 dilution of sample, use 9 milliliters of pure water and 1 milliliters of 1 to 10 sample dilution. To prepare a 1 to 1000 dilution of sample, use 9 milliliters of pure water and 1 milliliter of 1 to 100 sample dilution. 3. Once you obtain the proper dilution, use an adjustable pipette to place 100 microliters of sample dilution on a petri dish loaded with E&B agar. In our analysis, we use a 1 to 10 sample dilution. Use a spreader, pre-cleaned in an ethanol solution, to spread the liquid evenly on the dark surface of the petri dish. 4. Label and seal. 5. Repeat steps 3 and 4 on a new E&B loaded petri dish with other sample dilutions as desired.
In order to assure quality assurance and quality control, all data must be documented on field data sheet or within the site logbooks. All instruments must be operated in accordance to operating instructions as supplied by your manufacturer, unless otherwise specified in a work plan. Equipment checkout and calibration activities must occur prior to sampling, and they must be documented. In order to assure quality control measures, 1. A rinsate blank will be taken prior to sampling, and 2. A background sample will be taken at upstream from the sampling site.